Zorlukları geçtin, yeni evine hoş geldin. Hayatın 4 mevsim, sonbahar bu kez. Başlangıcı hayallerinin. Kış günü yarının yükü omzunda, ama yalnız değilsin bu yolda. Hazırlan bir çiçek gibi açmaya, kelebek gibi kanatlanıp uçmaya. Arılar gibi çalışmak gerek bazen, sonunda kavuşmak için yaza.
Good morning. My name is Nimet Ece 52. I am the Vice President of our Congress, and welcome to the Maltepe University International Student Congress 2022. I invite Dr. Uğur Baran Kasırga, the Vice Dean of Maltepe University Faculty of Medicine, for his opening speech.
I guess everyone hears me in good health and conditions, and I hope that there is no technical problem. Now, Excellencies, colleagues, member of Maltepe University International Student Congress, good morning. I would like to celebrate all of you in this heartwarming spring morning from Istanbul. It's an ultimate pleasure to be with you here today for this distinguished health summit. I'm honored and inspired by the spirit of collaboration among all young delegates that participate. Also, I am grateful to my dear friend, Dean of our Faculty of Medicine, Professor Selim Nalbant, who gave me the opportunity to address you now at this very critical moment in the global response to the COVID-19 pandemic that continue to impact so many aspects of our lives. He couldn't be with us this morning due to another international meeting he has to join, and I may pass his best regards and wishes to each and every one of you. Well, last 2 years, COVID-19 kept us all apart. COVID-19 has hobbled economies, strained societies, and unfortunately undermined the prospects of the next generation. They continue to suffer disproportionately from its socioeconomic consequences. Almost 2 years into the pandemic, a generation of humanity are enduring prolonged school closures and ongoing disruption to the health, protection, and educational services. COVID-19 pandemic is one of the most dangerous challenges of this world has faced in our lifetime, perhaps. It's above all a human crisis with severe health and socioeconomic consequences. While encouraging by the recent measures to reduce the incidence of COVID-19, we also recognize that this positive trajectory may not be predictable and sufficient enough. So this virus is unprecedented in our lifetime and requires unprecedented measures and responses. Obviously, in such conditions, it's possible that the same facts have had different readings by different entities. Once we have finally turned the page of this pandemic, there must be time to look back fully to understand how such a disease emerged and spread its devastation so quickly across the globe, and how all those involved reacted to this crisis. Now, the lessons learned will be essential to effectively address similar challenges as they may arise in the future, maybe soon. From this point of view, the contribution with your papers in this Congress is invaluable, friends. I took my chance to examine each and every one of your presentations individually and saw that Maltepe University International Student Congress in Medical Science and Nursing is playing a very crucial role in the global scientific community again this year. There are 25 in medical science and 10 in nursing sessions. Highly distinguished students' studies will be presented during the course of this Congress. From the new diagnostic trends of the COVID-19, clinical aspects of pandemic, treatments, drug and other therapeutic options will be available to the health hesitance, to the vaccine hesitancy. There are so many gems and are waiting for to be explored by the members of the Congress. Even the psychosociological aspects of pandemic on our lives, on educational systems and learning, teaching processes of medical education are covered. Now, dear distinguished members, in post-pandemic era, while each nation is struggling against the economic, social, and political crisis, like high inflation, deprived national wages, besides food and basic nutritional crisis, and lodging crisis of men, women, and children, there is also a darkest nightmare fact of bloody war and forced migration of innocent millions. In this summit today, we have a multinational participation from the countries like Ukraine, Russian Federation, Kazakhstan, Kyrgyzstan, Lithuania, North Macedonia, Romania, and Turkey, of course. At this moment, I believe it's very much important to underline, and best example would be the best example that how the positive science and the passion of researching unknown would be the strongest global glue available between the nations and the people. With this opportunity, I would like to thank in front of you for the each members of this management committee, students, colleagues, professors from all age and experiences who made this Congress available for us with their long work hours needed for that spectacular result indeed. Now it's time, it's our moment to meet and overcome the challenges of our time. We are stronger today than we are 2 years ago. Now, and we'll be stronger a year from now than we are today. I would like to thank one more time and each and every one of you for listening to me and being stars of this virtual audience today. Well, may God bless you all. May God protect all humanity from all the disasters. And thank you and wish all the best and very great success in your committee. Thank you very much.
I would like to thank Dr. Kasırga for his speech. Now, I invite Eylül Özköser, the Vice President of our Congress, for her welcome speech.
Dear participants, welcome to the 9th Maltepe University International Student Congress, which will be held online within Maltepe University. Human and Health in the Evolving World was chosen as the main theme of our Congress for 2022. 33 participants from 3 different countries will share about human and health in the evolving world during the Congress, which promises to be truly different and exciting. We have worked on this event with a great team of our instructors and friends. We are proud to be a part of Maltepe University Medical Faculty and MEDNS 2022. We are sure that our international This 2022 event will provide a wide range of experience and opportunities in scientific, academic, and professional aspects. We offer our gratitude to Hüseyin Şimşek, the founder of Maltepe University, to Professor Şahin Karasar, Rector of Maltepe University, and to Professor Betül Çotuk Söken, Vice Rector of Maltepe University and General Coordinator of Maltepe University International Student Congress 2022 for giving us the great opportunity to organize this international organization. We also would like to extend our thanks to Professor Dr. Selim Nalbant, Associate Professor Pınar Buket Thomas, Assistant Professor Çağrı Öner to support us during the preparation period of the Congress. Human and health in the evolving world is gaining importance in today's society. We are discussing a very broad topic that needs to be evaluated from different perspectives, like the increase in the prevalence of existing chronic diseases, complex diseases such as cancer, mental illness, and the biggest threat of the 21st century, COVID-19. In this Congress, we will point out how health affects people in our changing world with causes, effects, and consequences. This Congress will be a platform where students share their knowledge and experience on human and health in the evolving world. Following this Congress, we wish to obtain information about human and health in the evolving world, which are closely related to each of us, to create new approaches and to lay the foundations of new research ideas. We would like to thank everyone who has contributed and here with us today. We would like to wish all the participants a good time and greet everyone again.
Thank you, Eylül, for your speech. Now, I would like to invite Associate Professor Bilge Güvenç Tuna for her keynote speech. Associate Professor Dr. Bilge Güvenç Tuna completed her undergraduate education at Middle East Technical University, Physics Department, and her master's degree from Hacettepe University Health Science Institute Biophysics Department. She focused on smooth muscle biomechanics at Mayo Clinic in the USA. One of the world's leading investigation medical centers, Associate Tuna, worked on modeling of blood flow and pressure control, arterial mechanics, smooth muscles plasticity involved in the hip hypertension was observed. After completing her doctoral studies at Marie Curie School at the University of Amsterdam Academic Medical Center, Department of Biomedical Engineering and Physics in 2014, Associate Dr. Tuna has been working in Yeditepe University Faculty of Medicine Department of Biophysics since 2015. Dr. Tuna's recent research at Maltepe University focused on appomators, the development of targeted controlled drug delivery systems, the development of biosensor platforms, and the disease screening labs. The role of the vascularization, the epigenetics modifications in the effect of calorie restriction on aging-related disease. Dr. Tuna was deemed worthy of the UNESCO Laureate for Women on Science Award in 2019, the Science Academy Young Scientist Award Program in 2021, and the Turkish Academy of Sciences Outstanding Young Scientist Award in 2021.
Zorlukları geçtin, yeni evine hoş geldin. Hayatın 4 mevsim, sonbahar bu kez başlangıcı hayallerinin. Kış günü yarın çiçek gibi açmaya, kelebek gibi kanatlanıp uçmaya, arılar gibi çalışmak gerek bazen, sonunda kavuşmak için yaza. Zorlukları geçtin, yeni evine hoş geldin. Hayatın 4 mevsim, sonbahar bu kez başlangıcı hayallerinin. Kış günü yarının yükü omzunda, ama yalnız değilsin bu yolda. Hazırlan bir çiçek gibi açmaya, kelebek gibi kanatlanıp uçmaya, arılar gibi çalışmak gerek bazen, sonunda kavuşmak için yaza. Zorlukları geçtin, yeni evine hoş geldin. Hayatın 4 mevsim, sonbahar bu kez başlangıcı hayallerinin. Kış günü yarının yükü omzunda, ama yalnız değilsin bu yolda. Hazırlan bir çiçek gibi açmaya, kelebek gibi kanatlanıp uçmaya, arılar gibi çalışmak gerek bazen, sonunda kavuşmak için yaza.
Merhabalar, beni duyuyor mu herkes bilmiyorum.
Welcome, Dr. Tuna. I would like to introduce you, Dr. Tuna, for her keynote speech.
Sorry, is it going to be in English or Turkish? Because I thought it's in Turkish.
In English.
Oh, okay. Okay. Mm-hmm. So just a few minutes for the slides. Okay, I think now you can see my slides. Okay. So today I will talk about the smart drugs, about how they are used in medicine. I'm Bilge Güvenç Tuna from Yeditepe University. I'm an associate professor in biophysics department. And I'm working on targeted drug delivery by using nanoparticles. And there are many applications of nanoparticles in medicine and also targeted drug delivery. What do you get? By the way, Can they join or is it just one-way presentation? Can they ask for any questions or is it a one-way presentation?
You can invite any questions at the end of your presentation.
Okay, thanks. So the aptamers are basically used for targeted from 1990. This idea came up with the representation of silver bullet. So they suggested that if you can find a silver bullet which can directly target the disease area like a tumor, then you can solve the problem without giving any harm to the surrounding healthy tissue and also reducing the side effects. And starting from 1990s, aptamer antibodies, many molecules which has the targeting ability is started to use in especially in research, of course, and nowadays there are drugs that we are using especially for tumor treatment by using this kind of targeting approach, which are having FDA approval. So what are these aptamers then? As I told, there are antibodies. They are smart drugs because antibodies can, because I think as a medical faculty student, you know antigen-antibody relation. So if you have an antibody, then you can target any kind of protein on a cell or on an area specifically. And I am using another molecule, which is called aptamers. And these are having many different advantages compared to the antibodies to make the targeting. And these aptamers are single-strand DNA molecules or RNA oligonucleotides. And the first one invented, as I told, in 1990. And they are really promising candidates because also if you search the aptamer word in PubMed, you can see that it is day by day, the research related to the aptamer is increasing. And there is now in the market one FDA-approved aptamer. It's called Macugen. It's used for the neurodegeneration for eye. And that shows us these molecules are safe to use on humans. Okay, so why are we interested with aptamers? What are the advantages of them? First of all, their affinity, because since these are very specific small DNA molecules, their affinity and specificity is really high. What you do is you apply a selection process for a protein which is soluble in the drug, in the blood, or directly without knowing any specific protein, directly you can target the living cells. And also you can target inside the cell, outside of the cell membrane. You can decide and find a way to carry your drug the necessary point. And these are some representations of these aptamers. So as I told, they are DNA or RNA molecules, but they have also a 3D structure, and this structure is very important for their affinity and specificity. Another advantage of them are you can do in vitro synthesis, because when I say antibodies, I assume everybody knows antibodies. When you use antibodies for targeting, although they are very specific for their synthesis, you have to use in vivo environment. You need an animal to produce them. But aptamers can be produced in vitro, and that is also causing the advantage of easy to scale up, because you don't need live animals. That's why you can produce these synthetic molecules with large amounts. And also their size is really advantageous compared to, so this is an IgG antibody. It's very famous. That's why I have selected this one. Compared to this molecule, a thrombin aptamer, for example, is very small. So since we are using nanoparticles also conjugated with these antibodies or aptamers, because you have a target, to go next to the target, we are using antibodies or aptamers. That's why these are smart drugs. They have ability to find the target. And then you have to carry something there, because that's the job of these molecules is only targeting. Then you have the drug molecule that you want to carry to the necessary location. And to carry this drug, you need a package. And these packages are nanoparticles. And you load your drug into the nanoparticles, and you can add these antibodies, aptamers as a linker to, as like the writing the address on the envelope, these are finding the address. And then there are methods causing them to release the drug, the cargo molecule, on this necessary location. So that's why when you have a smaller molecule for targeting, that means you have still more place to make a total cargo. Your cargo can be larger if you have something smaller on it. So that's why the size advantage is very important for aptamers, definitely. And since you synthesize them in vitro, they are synthetic, their shelf life is very long, and you can store them longer time. They are more stable compared to the antibodies. And of course, since they are synthetic, the production cost is lower, and their thermal stability, pH stability, all kind of stability conditions are better for aptamers. So that's why we prefer to use aptamers as a targeting molecule. So what can we do with aptamers? First of all, how do we select it? You have a target. It can be whole cell, as I told. Maybe you don't know what are you targeting, but you can target, for example, leukemia cells only, because in the selection process, you find a DNA sequence or RNA sequence which can attach only leukemia cells and not to the normal cells. So that selection process lets you to find a specific marker without even knowing the marker on the cell. Or you can target cell membrane, you can target even a soluble protein inside the blood. And that's why aptamers are called optosensors. And usually they are conjugated with nanomaterials, and they can be used for diagnosis, for finding the cancer cells circulating inside the blood, for example. They can be used for the detection of intercellular diagnosis, and they can be used for pathogen detection. So they can be used as a biosensor, or they can be used for tissue engineering, bioimaging, and any kind of specific targeted therapy or theranostics. And the SELEX, maybe at the end of this speech, that will be the word that you will remember. Aptamers and the SELEX method, systematic evaluation of ligands by exponential enrichment. That's the method how we select these aptamers, because we should be sure that they are specific. And we, in my research, we use mesophorous silica in my group. We are using mesophorous silica nanoparticles. And they are also having many advantages compared to the other kind of nanoparticles. They have ordered pore network, homogeneous. They are really homogeneous in size. Their pore volume is high. That lets you to load more drug inside them. And their surface area also very large. So that means you can bind many aptamers around it to make the targeting. And you can easily functionalize. So all these modifications are called functionalization. So if you are chemically play around with nanoparticles easily, that means it's easy to functionalize them, because that's what we do. Normally, a nanoparticle is not, it doesn't have the ability to target something, but you are giving a function to it, and to generate, for example, it can, after this functionalization, it can pass blood-brain barrier. And then we perform controlled release. So what does it mean? Okay, you have the target. You are having the address of the target, like by using aptamers, or commonly antibodies are used. Then you loaded your cargo inside your drug. You loaded your drug inside silicon nanoparticles, but then you need a trigger mechanism to release it on the certain location. So it finds the location, but you design it to carry it. So there should be a signal to say this molecule release the drug. Otherwise, it will keep it in the circulation. So there are different kinds of controlled and release methods, but what we are using is aptamer gates. That's the novelty of my studies. So it is first, this kind of approach first used for ATP as a reversible gating. So normally, if you load without doing anything the drug inside the nanoparticle, then it will, when it finds its target, it will open there and release all the things inside it, all the drugs. But what we did is we applied aptamer, we put the aptamer as a gate. So it finds, aptamer has two functions in these studies. It finds the location, and also it's not releasing all the drug at the same time. It's keeping, it has ability to reclose the pore if the concentration of the drug is above a limit in the target area. So this leads you to keep the concentration for a longer duration at higher level in the target area. Because if it releases only one time, then it will release with a high dose at the location, but then it will be its activity will last soon. You will not have additional drugs which can be released in the location. So by using the gate system, we can keep the concentration at a longer duration. And there are different kinds of controlled release systems. This is like you can use magnetic field for the stimulation, for the opening of the gates. You can use pH control or a kind of internal stimulation. You know that in the cancer area, the pH is low or redox state is different in some disease status. So that low pH, for example, can be an internal stimulation for the release of the drug. Or external stimulation by using photons can be used. So pH responsive gating, thermoresponsive gating, and excitation-based gating are the most common methods for the release of the drug. So I don't want to dive into too much all the details of the aptamer gates. If you have questions, then we can go on. But I would like to show you some results. Like this is a study which we performed by collaboration with Associate Professor Pınar Thomas. You know her. So we used nucleolin aptamer, which is inside the phase 2 trials now. And we conjugated the nucleolin aptamer with silicon nanoparticles to carry the cancer drug carbendazim. And we showed that silicon nanoparticles are conjugated with nucleolin, and the carbendazim as a drug is loaded inside the silicon nanoparticles. And we showed that, first of all, the release kinetics. So we showed that when there is nucleolin in the environment, the drug is released and gradually released, not one at the same time. So in 24 hours, you can keep still high concentration at the location. And if it is inside the PBS, in a buffer, in blood, most probably, then it doesn't release the drug. It's keeping the drug inside. So first we showed this property. So it was working. The synthesis was working. And then we showed that it is more effective on cervical cancer cell line compared to the application of drug in a single way. So when you apply the drug with aptamer, then the effect was significantly more, and single drug did not kill significantly these cells. And also we showed that this is occurring due to apoptosis, and even the necrosis was increased, and the cell death was by these are the flow cytometry results. So one by one you can count the cells, and we showed that it is really working and more effective than the single drug. And these are additional figures for it, showing the strategy. And also, we did a similar study to show that we are really targeting in the body the tumor area by using animal study. So what we did is we generated a tumor model on a mice, and you incubated, you inoculate the cells to form the tumor on the animal, and after 2 weeks, the tumor formation is observed for breast cancer. And then we visualize while the animal is alive to see that if the drug is really reaching to the tumor area, or if it is just distributed all over the body or of the animal or not. And we saw that definitely after the injection, drug is only released on the tumor area immediately. In the body of the animal, you don't see any other fluorescence as you see. The fluorescence is only in the tumor area, and that is showing us that the drug is released only in the tumor area. And even after 1 hour, still that was the case. You don't see any other fluorescence image on the body of the animal. And also after 2 hours, still on the tumor, still after 3 hours, and it's also increasing as you see. It is accumulated more and more, and it reaches maximum level between 3 to 4 hours. And then after 24 hours, you don't see any fluorescence because most probably all the drug is used, and the silica particles and the synthesis that we did was removed from the animal, nothing left, and the animal was alive during all these processes. So I'm not sure about my timing. I don't know how much I can continue. Do we still have time? Or please warn me if to switch to the questions, because I would like to hear the questions more.
You can go on. We still have time.
How much
But if you
time do I have? Because I couldn't record when did I start.
around 20 minutes.
Oh, okay. Because I thought I have in total 40 minutes.
No, you can go on if you want. And if there are any questions, you can write on our YouTube channel. So I will going to lead you the questions.
Okay. That will be nice. There are other kind of applications for this targeted drug therapy. And as you see, we did many research on cancer, but you can use it for different kind of diseases such as atherosclerosis. Just a quick reminder of what is atherosclerosis is. It starts early, the progress is occurring throughout life, and it's usually goes on asymptomatic. And the atherosclerosis is the plaque formation on the wall of the arteries, and due to this plaque formation, the artery is getting narrow, and it can lead to obstruction, stroke, heart attack, any kind of end organ damage. And the main risk factors for the atherosclerosis is obesity. It can be something systematic or hemodynamic. Maybe from biophysics lectures, you know that there is laminar flow normally on the artery wall, but especially in the bifurcation areas, and also especially by aging, this laminar flow, laminar blood flow is disturbed. Sometimes it becomes turbulent at some locations, and causing the deformation of the endothelial barrier. So the systemic risk factors are usually the obesity, smoking, the genetic background, hyperlipidemia, blood pressure. But the hemodynamic, all these things are also adding on this hemodynamic risk factors, because On the bifurcation areas, where if you have disturbed shear stress, disturbed flow, it causes the damage of the endothelial barrier. And if this happens, this leads to a kind of inflammation, and the endothelial cells start to synthesize some chemokines to call macrophages to the area. And the LDL also can go under the endothelial cells, and the plaque formation process starts. And we know from the literature, especially, and also I have some studies on it, there is a molecule which is called transglutaminase. It works like a biological glue on the artery wall. And if you inhibit the transglutaminase, it really helps the inhibition of the plaque formation. It reduces the size of the plaques. So this is again from an animal study, and this is the carotid artery, which is getting low shear stress and oscillatory shear stress. And in the control case, as you see, the plaque area is really large, and you have many macrophages which is infiltrated to the plaque. And also in the oscillatory shear stress, as you see, the lumen becomes very small, and you have many macrophages. But when the TGT inhibitor is used, the plaque size is a bit reduced, and the number of the macrophages which are getting inside the intima is getting lower, and this helps for the treatment of the atherosclerosis. And but the problem is transglutaminase is a molecule which is used many occasions in the body. So you cannot systemically inhibit such kind of enzyme, and that is not a candidate of a drug, because it is, if you inhibit it, then you are disturbing many functions in the body. So targeted drug delivery is one of the best options for it. And that's why the transglutaminase, it has a key role in plaque formation. It should be inhibited, but you cannot inhibit it systematically. You should inhibit it locally. And that's possible if you have a target in that location. And we have selected ICAM1, a chemokine, for this purpose. And it is also very important for the monocyte attachment. And what did we do? Normally, you have the endothelial barrier and smooth muscle cells below it, and you have only the laminar flow on your arteries. But in bifurcation areas, if the LDL amount is very low, if the endothelial barrier is disturbed, then the disturbed barrier is causing more LDLs infiltrating under the endothelial cells. And during this process, we know that the ICAM1 proteins are expressed more in this area. So if you target these ICAM1 proteins to carry your drug, then these are your targeted drugs. And when they see the ICAM1 protein, they will also infiltrate under the endothelial barrier and release the transglutaminase inhibitor, the green one. So that's the strategy of approach for us. And we tried this strategy by using the cell culture experiments. And to mimic the atherosclerotic condition, we are not simply making a normal cell culture. We can apply shear stress on the cells, and we are really mimicking low shear stress, oscillatory shear stress environment to be sure that we are mimicking the atherosclerotic environment in the cell culture, and we are using pumps for it. And these are the HUVEC cells that we are using. We are using endothelial cells, and we applied normal, low flow, low oscillatory flow kind of different shear stresses on them to show that really we can do the targeting, and the efficiency of the inhibition, transglutaminase inhibition, was more. And we first, of course, select the specific ICAM1 proteins by using the SELECT method. And we found 45 DNA sequences. So that's the thing. you can find many sequences which are targeting this protein. But which one is functional and which one is the best? That's the real selection process. You should be sure that it is targeting this specific protein, and also it's not getting connected to other kind of proteins inside the body, because if it is not sensitive enough, selective enough, then it will bind also other parts. And this is not what we want. We want a specific targeting. And for this one, we use some biostatistics, and we also check their 3D dimensions, 3D formation from the by using the biostatistical methods. And at the end, we have selected 7 aptamer candidates, and still we need to find which one is the best. And that now we can go to the biology to test which one is the best. And to test it, we use first protein itself, and we have selected 3 candidates. And then we also did many experiments, so I will not dive into their details, but we showed that to the HUVECs, only one of them attaches the best. And then we characterize our nanoparticles, and this is all these characterization methods are showing their size. For example, in total, the cargo molecule was 177 nanometers. We showed that we can really attach the ICAM1 molecule on these silica nanoparticles, and we showed also the pore structure of these silica nanoparticles which are loaded with this drug. And as you see, it is 10 nanometers. So these pores that I'm talking are 2 nanometer size. So we are talking something really small but functional. And then we showed this cargo release is gradual again, so you can keep the drug at the constant level for long hours, like 24 hours, 48 hours. And then at the end, we showed that it is inhibited, the transglutaminase activity, more compared to the single drug application. So this is the control case, and the transglutaminase activity is green here. The blue ones are the nuclei of the HUVEC cells. And as you see in the third picture, you don't see much green. That means the transglutaminase activity is inhibited when we apply the drug with aptamer conjugated silica nanoparticles. And we did the same measurement for macrophages, monocyte THP1 attachment. In the control one, you can see the orange ones are THP1 cells, and the green ones are the cytoskeleton of the HUVEC cells, and the blue ones are the nuclei. And you can see in the control there are many attachments. Monocytes are attached on the HUVEC cells, but when you apply aptamer conjugated transglutaminase inhibitor, the attachment is comparable amount of less. And we did the same thing for LDL, low density lipoprotein infiltration. They should be oxidized. They are oxidized because when they infiltrate under the endothelial barrier. And we again showed that when you use the inhibitor, the infiltration is less compared to the single drug. So as a conclusion, you can select specific molecules on cells or in blood, like ICAM1 protein, to target the specific disease condition, and you can generate a cargo molecule by using nanoparticles and load this cargo with a specific drug. It might have many side effects, but if you have this approach, you can reduce the side effects and you can increase the local concentration of the drug in the necessary area while you are reducing the total concentration inside the blood. And we manage it for the inhibition of transglutaminase 2, and that is definitely prevented the monocyte attachment. So I would like to thank, because my presentation was a bit different. I wasn't aware that it will be in English, but I would like to finish with special thanks to Professor Dr. Doğan, which is in my team, Veli Cengiz Özal, and also Pınar Thomas for their contribution. And we have many PhD and graduate students, and I would like to thank all of them. Okay.
Thank you, Dr. Tuna, for her great speech. It seems like there is no questions.
No question. It's really sad.
Unfortunately, yeah. So I would like to thank you again for your honorable speech. We will and now we are moving on the student presentation. The first presentation is the first presentation about plasma therapy in patients with erectile dysfunction associated with coronavirus disease. The presenters, Solomia Prima.
Is my slide visible? Sorry, is my slide visible?
Yes, you can.
And are you able to see the movements of the slides?
Excuse me, can you repeat your question again?
Are you able to see the movements of the slides or no?
Yes.
Okay, great. Good morning, everyone. My name is Solomia Prima, and I'm a student of Ivano-Frankivsk National Medical University. I'm so pleased that I have such a possibility to be a part of Congress again. And today I'm here to present plasma therapy in patients with erectile dysfunction associated with coronavirus disease COVID-19. Actuality of our theme. Erectile dysfunction is an inability to achieve or to maintain erection, which is necessary for sexual intercourse. It is divided into psychogenic, organic, and mixed. Organic is the most common form, which is usually caused by atherosclerosis and also hypertension. The virus COVID-19 penetrates anywhere where blood vessels are and disrupts them through the specific receptors AC2. Penis is a highly vascularized organ of human body. Deficiency of endothelin, common in patients with severe course of the COVID-19, makes impossible starting erection. It deteriorates normal sexual life of patients. The plasma therapy of penis in combination with injections of prostaglandin and intracavernosous injections of phosphodiesterase inhibitors greatly intensify effect of these drugs. We aimed to research the development of erectile dysfunction associated with coronavirus disease, find risk factors of developing erectile dysfunction, and actually show efficiency and safety of using plasma therapy in combination with basic therapy. Research methods. We analyzed 15 sexually active patients aged from 35 to 65 years with expressed erectile dysfunction according to International Index of Erectile Function, which fell ill coronavirus disease and didn't receive sufficient therapeutic effect from basic therapy. We used International Index of Erectile Function and also pineal Doppler ultrasound. So the mechanism of erection. Erection is a spinal reflex that can be initiated by recruitment of pineal efferents, both symptomatic and somatic influences, from visual, olfactory, and imaginary stimuli. Nitric oxide is the central transmitter in this process. There are 5 stages included in erection, but we'll talk about 3 of them. First stage, filling with blood. As a result of psychological or sexual stimulation, impulses from the brain and nerves around the penis cause excretion of nitric oxide, and the smooth myocytes relax, allowing blood to fill in the corporal bodies. So we can see it on this scheme. Second stage, partial erection. Pineal arteries expand with the increased blood flow, which elongates the penis. And third stage, it's full erection. The increased volume of blood in the penis is prevented from draining by increased pressure, so it compresses pineal veins, expanding the penis to full erection. And we can see it on the image on the right side. So what causes erectile dysfunction? There is a wide range of reasons, but I'll give you the most important of them. 28% is psychological, from which the most common is depression, what is very relevant in post-COVID period. 22% heart and blood vessel disease, as we mentioned before, hypertension and also atherosclerosis. 31% type 2 diabetes, 5% hormonal disorders and injury to spinal cord each, and 9% surgery for bladder cancer. The mechanism of action of the virus COVID-19. Only knows that COVID-19 is a respiratory disease. It penetrates in human body with receptors AC2 and makes impossible touring angiotensin 2 in angiotensin 1,7. That is powerful vasodilator. However, what is interesting and what I want to say is those receptors contain in moderate amount in lungs and heart, while in testes, kidneys, and intestines they are in the greatest amount. So it damages testicular tissue and lowers testosterone level. It also damages endothelial cells and makes impossible to excrete nitric oxide, which is needed for relaxation of smooth muscles. Virus COVID-19 also damages parasites, which main function is regeneration. Plasma therapy is one of the best modern injection methods of treatment erectile dysfunction. Plasma is valuable thanks for its factors of growth, from which the most important are vascular endothelial growth factor and fibroblast growth factor. So it provides good regenerations of damaged by COVID-19 testicular tissues. We analyzed our patients before treatment, 1 month after treatment, and actually 3 months after treatment. Our patients received validated questionnaires, International Index of Erectile Function. According to their answer, we evaluated erectile function over the last 4 weeks. All patients complained of the inability to achieve and maintain erection, of sufficiency, rigidity, and duration to permit satisfactory sexual intercourse. So we evaluated our patients and classified them into 3 levels. Mild in 8 patients who had near 18.8 points, moderate in 5 patients who had near 12.93, and severe only in 2 patients who had near 7.86 points. According to results of penile Doppler ultrasound, before the treatment, men had highly reduced the peak systolic velocity, near 15.1, increased the peak diastolic velocity, 6.5, and resistance index was near 0.65. Patients who took only basic therapies, the values were peak systolic velocity 24.1, peak diastolic velocity near 5.1, and resistance index was near 0.81. And patients who had taken plasma therapy in combination with basic therapy during 1 month, the values were peak systolic velocity near 32.6, peak diastolic velocity near 4.9, and resistance index 0.85. And I want to share with you our final result, plasma therapy efficiency. 83% of our patients noticed improving of erectile function, but 70% of our patients unfortunately stayed without changes. And 63% of our patients after 3 months of using plasma therapy saved the results of it, while 37% still stayed without changes. And it looks like a small surprise or present for our patient. 36% of our patients noticed prolongation of sexual intercourse, in average near 4.7 minutes. Unfortunately, 64% of our patients didn't notice such changes. To sum up, virus COVID-19 persists in male genitals and it stays there for a long time. Patients 60 years old with chronic circulatory disorders are in group of the increased risk. Plasma therapy in combination with basic therapy improved erectile function in 83% of patients with erectile dysfunction on the background of COVID-19. Result of plasma therapy saved in 63% of patients after 3 months of using plasma therapy. 36% of patients noticed prolongation of sexual intercourse. According to regeneration disorders by damaging parasites, plasma therapy helps to speed up this process. So plasma therapy in patients with erectile dysfunction on the background of coronavirus disease is absolutely safe and efficient. And I also want to say, please don't forget about long-term effects of COVID-19. We must study and fight with them, and only in this way we'll change the world. So, thank you for your attention. Please take care of your health, be brave like a Ukraine, and I really hope that we'll meet next year. Thank you so much.
Thank you for your presentation. Now we are moving on with Ainur, which her presentation name is Evolving Methods in Health Research and Overviews on Zebrafish.
Hello, can you hear my voice?
Yes, we can.
Okay, I will upload the slides. Can you see the slides?
Yes, we can. You can start your presentation.
Okay, thank you. Hello everyone, my name is Ainur Genç. I'm a third-year medical student at Maltepe University, and I want to thank to the Committee of the Congress for this opportunity. I will make an overview presentation about the zebrafish today, which is a non-mammalian vertebrate animal model for biomedical research. For the past several decades, mice have been the leading experimental animal model for biomedical research. But its disadvantages, like their high husbandry costs and other certain limitations, lead to looking for alternative animal models. And zebrafish is one of them. Its genome is sequenced completely. About 70% of human genes have a counterpart in the zebrafish genome, and 84% of genes related to diseases again have a counterpart at zebrafish. They have several advantages over other vertebrate models, particularly for genetic mutant and therapeutic compound screenings. Aspects of human disease can be evaluated in vivo in zebrafish at the molecular level because of its highly evolutionarily conserved genes and signaling pathways. Its embryos are suitable for large-scale whole animal screening. The optical clarity of the larva allows visualization of individual genes throughout the developmental process using non-invasive imaging techniques. You can see the transparent zebrafish larva here. Embryo and larva are transparent in all zebrafish, but there is a certain strain called Casper, with transparent adult bodies as well. These are the zebrafish embryos on the right, again transparent. They are about to become larvae, and this is a closer look at the embryo on the left. There is a video, but I think I cannot play it here. I'm passing it. The advantages of zebrafish over the rodent models include their transparent tissue, high fecundity, rapid development, affordability, and easy maintenance. It is possible to perform toxicity testing, transplantation with few cells, genetic manipulation, and drug exposure on them. This graphic on the right shows the throughput and biological complexity ratios of research models. Zebrafish are more complex than in silico, in vitro, worm and fly models, and their throughput is higher than the mammal models. They somehow compensate for the certain disadvantages of the other models, although they have some disadvantages themselves too, like their difference in diet, microbiota, and environment from the human. And the scheme on the right again shows the advantages of zebrafish models. In the lab settings, zebrafish are kept in fish tanks, as you can see here. And this is the Nobel laureate, developmental biologist Christiane Nüssen-Volhard holding a zebrafish tank. She is one of the scientists who had used the zebrafish as a research model. Zebrafish models are compatible with new tools in research. Software tools like ZebraZoom to analyze animal behavior, a database for their genome, and tools for their cell evaluation are present. Also, they adapt to one of the most recent imaging techniques, which is the three-dimensional reconstruction of images to produce a 3D volumetric representation. Pathology in multiple organ systems has been studied with zebrafish. They exhibit a robust regenerative capacity in various tissues like their fin, spinal cord, and hearts. It makes them the sole regenerative vertebrate organism currently amenable for genetic manipulation. And future studies can enlighten us about the mechanisms of this regeneration and how to adapt it to the human body. Zebrafish have also been used to understand biological processes behind muscular dystrophy. Several mutants that affect muscle formation are identified in zebrafish, with one class showing a specific degeneration of skeletal muscle. Here is the evaluation of an aneurysm in zebrafish on the left. Since the aneurysm is a serious pathological condition of the human body, the ability to easily analyze it in an animal model with such clarity and resemblance is invaluable. And this is the evaluation of scoliosis in zebrafish on the right. Again, we can see the clarity of the analysis. The neuronal pathways of the zebrafish brain exhibit a profound resemblance to the human brain. Physiological, emotional, and behavioral pattern similarities between them also been well established. They are useful to study novel gene functions in neuronal development, and they can be used to study disease pathologies in Alzheimer's disease as well. Now I want to talk about the usage of zebrafish in cancer research. The field of cancer research has been innovative in last years. A trend towards targeted therapy and combination therapy has been facilitated by testing in various animal models in vivo. The murine model has been routinely used in cancer research. However, the main disadvantages of them is that it is impossible to study early tumor dissemination and the microenvironment. Thanks to the transparent Casper strain of zebrafish and its natural transparent embryos, it is possible to visualize tumor cell growth and dynamics at the early stages. Zebrafish cancer models can be created through the transgene expression and xenotransplantation. With these methods, you can see that several neoplasms, including thyroid cancer, hepatocellular carcinoma, and ovarian cancer, can be studied in zebrafish. Most tumor models generated in zebrafish are histologically comparable to the human tumors, and they possess important hallmarks of the cancer, like genomic instability and invasiveness. Here we can see the confocal microscopic evaluation of the tumor models in zebrafish. It is possible to study the growth of the tumor stages in detail. Tumor cell transplantation, xenotransplantation, is a relevant method for tumor invasiveness assessment. Tumor cells from a donor can be grown in a recipient or in another species. With small-sized transparent embryos lacking a mature immune system, it is possible to transplant and track high numbers of animals. In xenotransplantation, with the injection of the cancer cells, it is possible to investigate the growth, invasion, and micrometastasis of the neoplasms in zebrafish. As you can see here, it is possible to thoroughly investigate the progression of cancer and tumor angiogenesis with confocal microscopy in zebrafish models. With this method, patient-derived cancer cell xenotransplantation could lead to new options for personalized medicine. In one recent study, using a novel transgenic zebrafish model, 2 serotonergic antidepressants that suppress the hepatocellular carcinoma phenotype with beta-catenin-induced liver growth were detected. This phenotype is a commonly seen one for hepatocellular carcinoma. W-anti-beta-catenin pathway is a prominent parameter for many neoplasms. Here is the pathological and histological evaluation of the control group and the group treated with an antidepressant. And the graphics shows the suppression of cancer with antidepressant amitriptyline. Zebrafish is a convenient model for small molecule screening in drug development, owing to its high number of larvae at one place and time. And more recently, these models are highly considered for bioactive chemicals. Research at the subcellular level in zebrafish models again is widely used. They have been used as a preclinical model for the nanomedicines. The methods for this include creating gene-targeted mutation and stable transgenes or CRISPR-based approaches. These are the molecules used for procedures on the zebrafish genome, like chemical mutagens, viral vectors, transposons, etc. And TALEN, a restriction enzyme that can be engineered to cut specific sequences of DNA, has been overly popular. It's like the CRISPR-associated protein 9. And it was actually in zebrafish where it was shown for the first time that talons can produce heritable gene disruptions in the vertebrate genome. And there is a new exciting method, transgene electroporation in adult zebrafish, which was recently developed and used for site-specific de novo tumor initiation in zebrafish adults. With this technique, it is possible to inject DNA constructs containing tissue-specific promoters and genes of interest into adult tissue. This technique is invaluable as it is rapid and the expression of genes of interest can be controlled. And most recently, researchers have identified a new and unexpected kind of cell division that occurs without DNA being replicated, as we know as the normal cell division. Discovered in zebrafish skin cells, this new division, called asynthetic fission, could be a temporary measure used when growth is fast to make sure there is enough skin to go around. This discovery was made during research on the skin epithelial cells of the zebrafish. The scientists were using a multicoloring method to investigate the skin cells individually. You can see the multicoloring technique here with the individual skin cells. So in the skin cells, the division is highly rapid. With the absence of DNA replication and random splitting of the strands, many mistakes occur during this asynthetic fission. But interestingly, after a while from these rapid divisions, the cells produced through the fission are eliminated, and new normally produced ones replace them. These are my references and sources for the slides. And in conclusion, the unique features of the zebrafish models make them preferable to the rodent models in many aspects. They are promising for personalized medicine owing to the successful application of xenotransplantation and other new methods on them. And in an era when pandemics are on the rise, zebrafish, a model that can be used so widely in drug development, is increasing its importance. Thank you for your attention.
Thank you, Aynur, for your speech. Now we were going to move on with university pressure and socioeconomic factors, good predictors of somatic complements in university students. The presenter is Igor. Okay.
I'm not sure how well you can see me right now.
Yes.
The room I'm at is a little dark, but I'm confident you can hear me, right?
Yes, we can hear you. You can start your presentation right now.
So you can see it, right?
Yes, I can.
Okay, thank you. First of all, I would like to thank the committee of the Congress for selecting our. It's a great honor to be a part of your international Congress. And now I'll present the work that, along with my, which were Nina and Natasha, that we did. So it's entitled University Pressure and Socioeconomic Factors, Good Predictors of Somatic in University Students, with a question mark. So we were investigating that. So to introduction of what somatoform diseases are, These are psychiatric conditions in which the individual experiences various physical symptoms that are not rooted in any pathology. So usually these are predominantly very young or very old patients who are the two most vulnerable groups, who actually usually the patient history of this kind of profile, they usually wander from doctor to doctor because of a lot of these somatic complaints. The doctors, ranging from general physicians to internal medicine specialists, etc., try to exclude the diagnosis, but they find no evidence of an actual illness that has some organic pathology. So and yet the problem of the individuals persists. That is because the systems are often inconsistent with a possible physiological process, and the theory is not very well defined, but it's an assumption that some kind of maybe a stress or some other uh, stressor can actually create a side of the mind of the patient that actually manifests physical symptoms, and they are not in control of these symptoms. And it's very important that the specific disorder that we actually try to examine with this study, which is somatic symptom disorder, the people can control the symptoms, and they do not have fake symptoms, such as in fictitious disorder, maybe. And it's also different from hypochondriasis, where people actually are inventing symptoms that are not there, which is very important to note. And a very important uh, point to make is that the symptoms pass only when the physiological factors that lead to the symptoms are resolved. So when the psychological conflict that persists in the mind of the patients ends, so do the symptoms. And it's usually a very abrupt process. When the conflict ends, it's very abrupt, not gradual, like you would expect with an actual chronic somatic illness. There are a few factors that can actually predispose to the formation of somatic symptom disorder. I mentioned the risk groups, who are the very young and the old, because they are usually more prone individuals to stress, who have not built up that life resilience, and that comes with experience. And the prevalence varies across many studies, and it's different and across the different populations on the planet. So the number one risk factor is increased life stress, also previous history of depression, ongoing anxiety or depression, family history of somatic, which is very important because a lot of the pathophysiology of uh, the somatic symptom disorder is rooted in a genetic basis. And also very interesting, having an actual medical illness can predispose to having these somatic symptoms, which are not actually related to the actual disease, but are completely unconnected to it, and are usually organic systems of the organism of the patient. When taking into account today's fast lifestyles and various other environmental stressors that did not exist, let's say, 50 years ago, the prevalence of these disorders has been increasing consistently in the last 25 years. But it's very important to note that despite this, just like a lot of diseases and illnesses and many psychiatric conditions, a contributing factor to the increasing prevalence might be the fact that the diagnosis competence of the doctors or the psychiatrists that are disorders has increased with time. So the aim of our study was to examine the prevalence of somatoform disorder and symptoms among university students in the St. Cyril and Methodius University in Skopje, and try to connect these somatic symptoms to various other aspects of the participant. For that goal, we created a Google Forms question, 15 questions, which were part of the PHQ 15, or the Public Health Questionnaire, which is a diagnostic tool or questionnaire which is used to examine the prevalence of somatic symptoms in individuals in the last 4 weeks. There are a few 15 questions, and then we had an additional 20 other questions which pertain to the various other and factors in the life of the participant, excuse me. So this is the PHQ 15, and as here we have symptoms such as stomach pain, back pain, extremity pain, some menstrual cramps, which is a condition only in women, headaches, chest pain, dizziness, fainting spells, racing of the heart, shortness of breath, pain or painful sexual intercourse, constipation or diarrhea, indigestion, feeling tired or having low energy, and trouble sleeping. And for each of these, the participant can answer whether they're not bothered by the symptom, whether they're bothered a little bit, which is scored a 1, and bothered a lot, which is scored a 2, and then these scores are summed, then you have the final PHQ score, in which a score between 5 and 15 indicates mild somatization, between 10 and 15 moderate, and over 15 severe. So the condition in our study was that somatic symptoms were actually every participant that had a score over the various other questions, which were 20 in total, pertain to the age of the patient, the gender, previous history of somatic employment status while studying of the participants himself or the participants or parents, the performance at university in accordance to one's expectations, the on university studies that they place, history of depressive disorders, anxiety disorders, family history of somatoform illness, regrets regarding the chosen university major, perceived career prospects in the home country or the hometown, the level of difficulty of the university curriculum, whether they were far from home, whether their hometown was very distant from the capital in Skopje, are they an only child? They have a regular social cycle? Do they generally feel stressed out at the end of the day? Do they get hours of sleep per night? And their smoking status. The questionnaire was distributed to students via the various social and online communication platforms from the 17th of February to the 20th of March 2022. So that was Viber, Messenger, and WhatsApp. Then the pooled statistics, or the data that we had, was analyzed. First, descriptive analysis was performed. Then the participants were divided into 2 groups: those who had a somatic score on the PHQ 15 over a 5, and those who had below 5, or that was the control group. And then an odds ratio, p-values, and confidence intervals were calculated by comparing the results of the 20 questions with the score of the PHQ 15. The study had, excluding incomplete surveys had 181 participants. The gender breakdown was that 118 of all participants, or 65.2, and 15 or 60% of all participants experienced somatoform symptoms were female respectively. So there was a huge bias towards gender in the cohort, and the average age of participants was 22.7 years. We didn't include the descriptive statistics in this presentation because there was a lot of data for all 20 questions. It would be pointless to prolong the presentation. A few things that were striking about it was that there was a high percentage of smokers cohort, almost 60%, and almost a third had some kind of job while being at university. So in the somatoform disease group, or those who had a score over PHQ 15, there were 25 participants in the study out of 181, or 13.8%. The breakdown was that were experienced by 14 individuals, or 7.7%, moderate symptoms by 11 individuals, or 6%, and severe. There were no participants with severe somatic symptoms. Out of all of the questions from the survey, or the questionnaire rather, they were calculated for an odds ratio, a confidence interval, and a p-value. The significant p-value was determined at below 0.05. So only 4 parameters met the criteria, so only 4 had a significant correlation. Those were underperforming in university studies according to one's self-expectations, with an odds ratio of 4.09, a confidence interval of 169 to 99. The next on the list was previous history of somatic complaints, odds ratio of 3.73, and a confidence interval from 154 to 903, having a job while studying that had a odds ratio of 30.33, a confidence interval from 1.4 to 7.93, and placing very high importance on university studies in one life with an odds ratio of 2.75, and with a range in the confidence interval from 1.16 to 6.54. There were other parameters which show the odds ratio, or that's a odds ratio over 1, such as the smoking status, the unemployment status of parents or guardians, but they had a insignificant p-value, so we excluded those statistics. There were several limitations of this study. First, it was a self-reported study, so there was no analysis by a skilled clinician who could determine the symptoms were real or maybe fictitious. It was an online study which required identification. Only university students from the university in Skopje could participate, but it was also it could be, it's not reliable when you have an online study. It's not as reliable as conducting a face-to-face interview. There were a majority of 65%, which could be biased because, according to some studies, women have a higher incidence of somatic symptom disorder, and there were students from only one university. That was the St. Kirill and Methodius University in Skopje, which could be a bias because Skopje is a very big town, the largest city in the can contribute to more stress in some predisposed individuals. So to make a summary of the results, work significantly correlated with somatic symptom prevalence in this study. So an abundance of stress and responsibility could contribute to somatophore manifestations in these prone young individuals, also striving for high academic achievement and also failing to perform in accordance to that expected academic achievement were correlated to somatic symptom prevalence. This could be more personal, not just because of the stress level, the general anxiety it can cause, but also because, in a broader sense, it ties into the personal identity and future career prospects of that individual. And the last significant correlation was between previous history of somatic symptoms and current somatic symptoms, which in the literature is clearly defined as one of the biggest risk factors for current somatic manifestations. And also these individuals have early current symptoms throughout their life. So to conclude, our study determined that almost a 6 or 13% displayed significant somatic symptoms among the students in the St. Cyril and Methodius University in Skopje. Despite this, especially in younger, these symptoms can be seen as banal, hypochondriac complaints, even by clinicians, because these are healthy individuals in the prime of their health, but yet they feel very sick and very like they have some bodily disease that actually impairs their everyday functioning. Psychosomatic disorders have a high burden on the social and professional functioning of the sufferers, which can reduce everyday performance and general well-being, which prompts a more careful approach by clinicians when potentially encountering these individuals in their everyday practice. And we would like to end this presentation. Set peace of mind as your highest goal and organize your life around it. So that should be the priority for everyone, not just these young people we included in the study. Thank you for your attention, and thank you for allowing us to be part of your Congress once again.
Thank you, Igor, for your presentation.
Thank you.
Now we are moving on with the other presentation, clinical case of infertility with large uterine fibroids, presented by Momin Mohammed. Okay.
Good morning, all of you.
Good morning. You can start your presentation.
So my name is Momin Awais. Yes. My name is Momin Awais. I am studied in Kyrgyzstan Medical Academy. And today I represent my presentation, that is clinical case of infertility for large uterine fibroid. So first of all, we see what is leiomyoma. Leiomyoma are benign tumor which originate in the smooth muscle cells of myometrium. And uterine leiomyoma is one of the most common benign tumor of the female reproductive system. An important role is to assign a gynecological pathology. First of all, we see a clinical case regarding this condition. A patient, 37-year-old, complaint of enlarged abdomen, aching pain in lower abdomen, and difficulty in urinating. First time, a small size uterine fibroid diameter 2 centimeter is diagnosed 7 years ago when patient is observed by infertility. And past 6 years ago, the patient is not observed by any gynecologist, but in 4 months ago, patient is observed by a gynecologist because the patient is suffering from abdominal pain and lower abdomen difficulty in urinating. So we diagnose a subsequent intramural uterine. So we plan for the treatment, and according to the condition, the patient is advised to conservative myomectomy. We conserve the uterus and also we remove the uterine fibroid. So before we plan the treatment, we plan the treatment with three methods, that is pre-operative, operative, and post-operative. And in pre-operative treatment, first we see the uterus size is fibroid size is very large. So we use a GnRH agonist therapy. And the use of GnRH agonist therapy is that GnRH agonist reduce the size of the uterine fibroid and volume, and is very helpful for our operation and change in hemoglobin. And in intraoperative effect of GnRH is duration of operation and loss of blood decreases and proportion with vertical incision. And in post-operative effect of GnRH is complication duration of hospital stay. Then the patient is staying 6 days, 7 days. It reduce the staying. So first we see the result of preoperative preparation. When we not use the GnRH, the size of the uterine fibroid 11-10 centimeter cube, and after we prescribe the GnRH therapy, the size of the uterine fibroid 10 centimeter cube. So first we see the what is GnRH therapy work. We all know the GnRH is useful for released by the hypothalamus and it stimulate the pituitary to secrete FSH. And when FSH is released, the menstruation is occur and ovum is released. But if we continuous give the GnRH therapy, it act like GnRH therapy, invasive GnRH therapy. So the size of the uterine is decreases, uterine fibroid decreases. So after we see that we consider a myectomy performed and the fibroid was successfully removed and the uterus was preserved. You see the size of the uterus, and if we not use a GnRH therapy, we see the blood loss. If we do not use GnRH therapy, the more blood loss and patient is suffering from anemia. And after the post-operative treatment, the post-operative period was uneventful and the patient received antibacterial therapy, because if any bacterial happen, then we use antibacterial therapy, uterotonical therapy, anti-anemic therapy, because if any anemia happen after operation. And adequate pain relief therapy we give the patient if any pain happen. And prevention of thromboembolic complication like endometriosis. And after the operation, we recommend the patient that is continue a GnRH therapy for 1 menstrual cycle and Dino just 2 mg per OS according to the scheme of the GnRH agonist for 5 months. The function of Dinogest is like synthetic progesterone, and progesterone prevent pregnancy, because you see the after operation control ultrasound for 1 month after operation, and planning for pregnancy after 6 months. Why we planning pregnancy after 6 months? Let us see. Because if we planning for pregnancy earlier, so at the time of operation, we see the fibroid is half is inserted in endometrium and half is outside the endometrium. So at the time of operation, when we remove the fibroid, the endometrium is also damaged. So the skin, if the skin incision is healed 1 month, or 20 days, but the endometrium is healing time is healing is 5 months or 6 months. And when we do planning for pregnancy, it is chance for miscarriage, and our purpose is not achieved. So first of all, we tell the patient for planning for pregnancy after 6 months. So post-operative period, so the healing of the surgical wound is occur properly, and is not occur properly, our purpose is not achieved, because our main purpose is to make the woman fertile. So after we see the ultrasound, so we check ultrasound after 40 days of surgery, and before we see before operation, the size of the uterus is enlarged up to 16 months of pregnant woman. Like then the after operation, the size of the uterus is normal, and the centimeter is 67 and width is 56 and anterior posterior size, and the contracture are even, and the echo structure is homogeneous, and the cavity is clean, and thicken of the endometrium 6 So in conclusion, we see the presentation, I present the clinical case is example of the best management of patient with uterine myoma in combination with infertility. Because in this case, we see the women no problem in organ, no problem in uterus, no problem in ovaries, because main problem in fibroid, and the fibroid is not become the woman fertile. So our main purpose is remove the fibroid and conserve the uterus. And if we don't use such therapy like intelligent and our clinical skill, so we remove the fibroid and uterus is also damaged, and no purpose, and woman be not fertile. So our main purpose is to become the woman fertile. So as a doctor, when we study MBBS or any other courses, so please See, you use your clinical skill properly after operation, because if you see, if you don't use DNRH therapy. So if you don't use DNRH therapy, then see the uterus cut section of the fibroid. If you don't use DNRH therapy, the patient is suffering from anemia and large of the blood is lost. So if you use DNRH therapy like a clinical skill, you use suffer patient is not suffering from anemia and you operate properly. And the relation for the use of pre-operative medical therapy before surgery for fibroid is to make surgery earlier and the woman become fertile. Thanks for your attention.
Thank you for your presentation, Momin. Now we are moving on the other presentation. Name is COVID-19 Effect on Medical Education by Nawal.
Hello.
Hello.
Hi, Nimet. Can you hear me?
Yes, we can hear you. You can start your presentation anytime you want.
Thank you. So today I'm going to be presenting the impact of COVID-19 pandemic on medical education worldwide. All right. So as background, we will be talking about the COVID-19 pandemic led to major changes and instabilities to medical education across the world. We assessed medical students' perceptions of training, their experiences, and alterations in instructional approaches during the pandemic. We used methods like our online survey that was conducted among a various international network of medical students. Students were surveyed on their perceived overall impact of COVID-19 on their training, as well as other exposure factors. To establish the strength and associations, univariate and revised analysis were carefully performed. First, we were able to obtain the ethical approach from our survey, people who took our survey. Prior to participation in the study, all individuals signed a written consent form. Our results included the following. We collected several complete questionnaires completed by medical students from 8 medical schools around the world. Participants were predominantly female, about 67% female, and 33% male participants. The median age was 21. 12.5% of the participants reported that they suffered from financial problems related to the COVID-19 pandemic. Participants reported from various levels of study. They were either doing their studies in medical school or doing their studies in clinical training. Almost half of the total is in their clinical years. The majority of the participants, about 79%, said that they were very competent or adept at utilizing technological gadgets like our online learning sessions. About 66.7% of the participants reported that their faculty suspended or postponed the educational program due to the COVID-19 pandemic. Our results included the following. Half of the respondents, 50%, said they work in a hospital as physicians or interns in training as part of their educational program as a student. About 32% reported their concern about being exposed to COVID-19 during their clinical practice or fulfillment of their duties at the hospital. About 55% reported that they are feeling more anxious now than they usually do during their studies as a result of the COVID-19 pandemic. They also reported that medical information regarding the pandemic was requested from them by family, about 54%, and friends, about 29%. The use of social media to disseminate information about the COVID-19 epidemic was widespread, according to our participants. More than 50% of our participants disagreed that e-learning is a possible substitute for standard medical education. Conclusion. According to an international survey of medical students from around the world, which is the one we performed, the COVID-19 epidemic has had a substantial detrimental influence on their medical education. While there has been a considerable increase in the usage of virtual learning techniques throughout the world, more study is required to evaluate the validity of the goals set for these teaching tools. So as a result, I want to conclude in that the main purpose of our research and study and questionnaire was to fully be exposed to how medical students are feeling regards the COVID-19 and how they were perceiving its effects on their medical education worldwide, which is why we also surveyed the students in different countries as well. Thank you so much for attending and have a good day.
Thank you for your presentation, Nawal. Now we are moving on with the poster presentations. Now the students' presentations are over. At the end of the poster presentations, our medical session is over. After our medical session, nursing session will be starting at 1 p.m. So we see you later. See you tomorrow on the other sessions of medical.
Zorlukları geçtin, yeni evine hoş geldin. Hayatın 4 mevsim, sonbahar bu kez. Başlangıcı hayallerinin. Kış günü yarının yükü omzunda, ama yalnız değilsin bu yolda. Hazırlan bir çiçek gibi açmaya, kelebek gibi kanatlanıp uçmaya. Arılar gibi çalışmak gerek bazen, sonunda kavuşmak için yazar.